Purser, Dan Purser, MD.com here. And we've got a special guest for this podcast, this Facebook Live today. And his name is Rick Martin. He's the CEO of MicroGenDx. You guys have all heard me talk about this a number of times. It's a great, great product. I was a microbiology major at BYU and was stuck in the culture world like a lot of doctors are. What a nightmare. And I found this technology and have loved it. It's saved a lot of you, a lot of misery, and helped a lot of my patients. It's an easy, wonderful technology. And I'm going to quiz Mr. Martin here, who's, like I said, the CEO of MicroGenDx, on this really cool new technology. We're finding out what's causing your infection, what's causing that misery, what's causing that bladder problem that won't go away, what's causing all this stuff, what's causing that sinus infection you've had 4 years. I had one of those myself. Yeah. So this is Rick Martin. How you doing, sir?
I'm doing great. It's a pleasure and honor to speak to you today.
Yeah, no, it's super cool. Thank you for giving us the time. I know you're incredibly busy, so and I can relate to that. So, but I've been wanting to do this Facebook Live with you for a while. So, so I've got a set of questions I want to ask you. I hate to put you on the spot, but I thought these would be helpful. And later, if you want to question me, you certainly can flip it. So we'll let you do that because I love your product. Thank you for being involved with this company and helping them get it out there. So
I didn't realize you had all that experience with it already. That's great to hear that, because usually we have it, especially people with microbiology background. It's even more difficult to change people that have the micro background. So that's great to hear.
There's still a little place for microbiology in the world, but
Yeah.
at least in infectious disease world. And that's not really my specialty. For 30 years I dealt with complex wound issues, and this would have been really handy to have for that. So it should be looked at hard for that. So what is this we're talking about? What exactly is next generation sequencing, and is it PCR? What's the difference between these?
Yeah. So, one thing I guess we can thank COVID for is it's educated the world on what the term PCR. So, and before COVID, probably, most of the population didn't have any idea what a PCR was, but, and now we're throwing another term in there called next gen sequencing. So it's a question I get asked a lot, and essentially, PCR is a bias test in the sense that it goes out and looks for whatever you ask it to look for. So from the COVID, we know there was a set of genes that were within the COVID virus. So you could build primers, which were basically a way of looking for that specific gene, and a PCR machine could rapidly tell you within hours whether the gene was there or not, because it's only asked to look for that one or two genes. So PCR is biased to what you ask it to look for and what you set it up for. If there were other things in there, let's say, well, I don't know, because I don't have a primer to go and look for that.
Right, visible to this test, yeah.
Yeah. So next gen sequencing is unbiased. So it doesn't really care what genetic material is in there. We extract all the genes, all the DNA from the sample, and like every person has its own unique DNA, every microbe, every bacteria, every fungus has its own unique DNA. And so if we have a database or a library of what we call the sequence codes for the DNA of those species, then with next-gen sequencing, we can take a sample and we can extract all the DNA from the sample, whether it's urine or sputum or bone or tissue. We can extract it, and then we can match it to a database or a library. So we can say with, precision, like literally 99% precision, that what microbe is in the sample. And so it's essentially, the analogy I like to use is like the crime scene. People watch CSI, and they collect DNA at a crime scene, right? And then they take it back to the lab, and then they extract it, and they basically have to have a library to match it to. So if you're in the FBI's National Library of DNA, and they have your DNA from the crime scene, they can match it to the library or the database and say, well, you were there. And essentially, that's what next-gen sequencing is now. We can have a massive database. In our case, we have 57,000 bacteria and fungus in our
Huge.
database.
Yeah, you have
So
huge. Yeah, it's huge, monstrously huge what
yeah,
you guys have.
yeah.
So
So PCR, PCR is put into what we call panels. You would never have a panel of 57,000. So that's really the difference, is that you don't need these limitations of biasness of a panel. It's unbiased. It's gonna pull the DNA and then match it to a database. And the bigger the database, the better curated the database, the more accurate it is. And so that's essentially what's next gen sequencing. And so the barrier to this was cost and time. It would, take a university a month to sequence a sample, which is not very practical for clinical medicine and making decision making.
right.
And cost was the other barrier. That those are both the areas we've kind of addressed.
So I know my followers are going to have a big question about the PCR testing. You're really doing PCR level 2 testing, right? Is that correct? Or yeah, where
Yeah,
you're the next gen sequencing.
yeah, yep.
What their big concern would be is, well, does their DNA get picked up, and how do you deal with that? I mean, what that, because
Yeah,
they're all worried about privacy issues. So
yep. Get that a lot too in questions that, yeah, patients will call our customer service center and, raise that concern. It's like, we're not looking at the host DNA, your own DNA. We don't extract that. We are only looking at the bacteria and fungal DNA. So we have no way of being able to pull your DNA and put your DNA in a database. So we're strictly looking at bacteria and fungal DNA and some viruses.
that's awesome. That's the number one thing I found I have to overcome, because everyone's like, wait, will this give my DNA? And I'm like, no, I don't think they even look for it. So I was saying the right thing. But what you do find, which is really cool, and is that you find out the resistance pattern of some of these bacteria and funguses. Anyway, yeah, I didn't mean to bring that up, but I think that's a really cool aspect of your technology. That's really neat. So,
Oh yeah, for years when we've been around since 2008, and in the early years when we were doing next gen sequencing, we couldn't tell the physician whether the patient had resistance to common antibiotics like quinolones or aminoglycosides and so forth. So,
yeah.
and as a physician, you have to have that sensitivity data. You say, okay, you got a bacteria, but I need to know what's it sensitive to. And so, one way around that is we do use PCR to be able to design the specific primers for those genes that cause resistance. So we do a two-step process. We run the sample through looking for those specific resistance markers or genes, and be able to tell the clinician, hey, this patient's urine has quinolone resistance. So don't use Cipro because it's probably not going to work. And then here's the bacteria that we found or the fungus that we found in the sample. So yeah, that's important to get that. It's in traditional culturing called phenotypic sensitivity, where you grow it and measure it, how it responds to the antibiotics in terms of, there's different methods of how they do that. But we're looking at the genotypic or genetic resistance markers for antimicrobial resistance.
Super cool. I'm gonna get ahead of myself a little here, but three aspects of your product that I really like is the price. We'll get to that later. I'm gonna quiz you on that later. Um, um, that resistance factor and the speed you guys get stuff done. It's really out there. I mean, it's really neat. So compared to 3-4 weeks waiting for culture and sensitivity to come back and dawdling, patients getting worse, and you're just guessing, hitting them with different antibiotics, it's a nightmare. So what you guys have done is kind of smoothed all that out and gotten it out of the way. It's really cool. So thank you for doing that. That's super neat technology. So the next question I've come up with, why is it important for people to understand what bacteria species or fungal species is causing their infection?
Well, I think it's really important that people say, well, why do I care? Don't I just give me an antibiotic? But if you have a respiratory infection, as a physician, they'll say, well, I just need a Z-Pack. Give me a Z-Pack. And that worked last time. or if I have a urinary tract infection, I was on Bactrim last time, give me Bactrim. And so I think the general population just perceives the physician needing to write an antibiotic or an antifungal. But as you know, the reality is we need to know precisely what type of bacteria, because not all antibiotics work against all bacteria, and not all antifungals work against all fungal species. So I think it's important that the general population understands that the first step in diagnosing your infection is making sure we understand what species or multiple species are there causing you to feel miserable. And so if we know that information, then we can prescribe the right antibiotic. Versus medicine really right now is because of the system is forced to treat empirically, which is essentially guessing.
Yeah.
If you come into your doctor's office and you have United Healthcare Insurance or Humana, and they're contracted with LabCorp and Quest as your network labs, and they're going to send off a culture, and if in a UTI, 60% of the time it comes back as mixed flora or nothing. And so what does the doctor have to do? Well, they're going to treat empirically. They're going to say, well, I've seen this in the last 10 times, and this antibiotic worked eight out of 10 times. So I'm going to go with that antibiotic. And so that empiric approach is not the best in terms of patients who it doesn't work. And now they're miserable, and their symptoms go on, and they're at risk for hospitalization. They're at risk for antibiotic resistance, which is a huge global problem that we're facing, because the antibiotics, because we don't know what we're treating, we're just blindly throwing antibiotics at them, and then the bacteria love that. They'll just say, okay, I can learn, and all of a sudden they're resistant. And it's really amazing. We have more than 200,000 urine samples in our database, and a lot of women that have recurrent chronic UTIs. When I look at the reports, and we're going to publish a paper on this, but the amount of resistant genes is just unbelievable, and it's growing.
Yeah, it is.
Because they're just being shotgunned with one antibiotic after another, and then the end result is they have multiple drug resistance and microbial resistance, and that's not good.
Interesting. And when I first found you guys, I cultured didn't culture myself. I used your technology. Yeah, I know everyone says that. I used your technology and got a sample from my sinus, just put it up in my nose. I'd had it for 3.5, 4 years, thinking I eventually thought, do I have throat cancer? What do I got? It was so mild to begin with, and then it just became a problem. I'm like, I have a sinus infection, and I couldn't figure out what to treat it with. So I did your technology, your testing, found out I had 5 different bacteria. I'd been state medical director and gotten all kinds of weird, when I in and out of ICUs and nursing homes, got all kinds of weird bacteria in there. And it took me about 3 testings to clear all of them out. It's like peeling the onions, what I explained to the patients. So it's really cool, because I found the 5, then I found 3 more, then I found 2 more. It's just a cesspool in there, horrible to think about. So, and then some people have to do that. We do that same technology, that same pattern with interstitial cystitis cases. It's like peeling an onion, sometimes trying to figure out all the infections they've got going on in their bladder. So I love that, what you just said. You hit the nail on the head with that. So thank you. So are infections caused by, I think we know the answer to this. Are infections caused by a single bacteria or single fungal species? Yeah, sometimes. But what do you think about that?
Yeah, I think bacteria behave in 2 ways. They for acute infections, it's often what we call a planktonic bacteria, or a single bacteria that's causing the acute infection. Chronic infections are often result of a community of microbes coming together and forming what we call a biofilm in many cases, where they just become it's a polymicrobial, and they're working together. And so you really you need to understand the whole community that's there. And it's the challenge of our technology is, as you probably saw, is interpretation, because all of a sudden you're going to be seeing bacteria you've never heard of, because only 1 to 2% of all known microorganisms will grow on that petri dish or that traditional culture.
Right.
So
Exactly. Oh yeah. Yeah.
it's really missing a lot. And if the bacteria
And your
go into,
technology doesn't miss, by the way. That's one thing I
yeah,
tell the patient. It's going to
yeah,
find everything in there, whether you want it to or not. There's things
yeah.
hiding in there. Okay,
Yeah,
sorry.
the challenge comes when we take a sample from an area where we have a microbiome. So we know our body has, as many bacteria cells as host cells. So it used to be, if they thought was there was 10 to 1, but it's really probably equal. But there's still a lot of bacteria in us, and we need it in our gut, we to survive. We have it on our skin, we have it in our sinuses, we have it in our urinary tract. Medicine used to think, and I had a doctor call last week that says, what do you mean? The urinary tract is sterile. I was like, no, it's not. Everybody has microbes in their urinary tract. It's not sterile. And so when you get an infection, that community of microbes that's living in there and they're living happily with you as the host, and then all of a sudden, like maybe you have sex, and all of a sudden a microbe from your partner comes in and now changes the community in the urinary tract. And the body then says, hey, these guys weren't here before and we don't like it. And so then comes the whole inflammatory response, the white blood cells, the inflammation. And so part of our challenge is interpretation of the report, because there are going to be some microbes that are going to be there that were there before. And so in the future of this technology is going to be that you could take a baseline of your own urinary tract microbiome. Say, I feel great right now. And so let's take a sample and let's see. Like any lab test, you always see in your lab reports, you always see what's the range and what's my value. And so this is very similar in the sense that if you're taking a sample from an area where we know microbes live, like your urinary tract, like your sinuses, that you can take a baseline measurement of what is there when you feel good. And then when you feel bad and you have inflammation, take another sample and say, oh, how did the community change? Well, these species weren't there when I was feeling good. So maybe we target those. Or the easiest way too is like your sinus example. I'll tell rhinologists, you're going to put a patient on antibiotics, you're going to do steroids, you're going to do a FEST procedure, surgery to clean her out. All those things are disrupting your normal microbiome in your sinuses.
Yeah. Oh yeah.
And so when you look up in the patient's sinuses and you see inflammation, well, that tells you there's somebody still causing inflammation. Take a sample and find out who still survived. What microbe is still there after everything you've done? And that's the target, because all of a sudden you're going to see that the community has been disrupted, and now maybe there's only one or two bacteria that are just dominating that space, and that's your target. So it's a learning process that medicine has to go through, but we have a lot of doctors who get frustrated with the interpretation of the reports and they just kind of give up. It's like, no, you can't. This is not going away. This is the future, and it's here now. You have to learn how to interpret it and make these clinical decision making.
I like that point. It is critical. One of the things that your company offers when you're really scratching your head with a patient result is you can talk to an ID, you can talk to an infectious disease guy or gal, and they'll guide you. I've already done that a couple of times. It's been really helpful on some of these issues to go, I just don't know what I found here, and how do I treat this? So yeah, it's been good. I get a lot of weird stuff. My patients, I'm a cash pay practice, so my patients come in, they want service, and and this test is a bargain to them, no matter what anyone else thinks. It's cheap, I think, for the technology you bring, and really inexpensive. I shouldn't call it cheap, but quite inexpensive. But yeah, the fact that we can get an infectious disease, essentially console on the backside if we need it, is really handy, really helpful. Yeah. So okay. So what kind of indications is this test good for?
Well, as a high complexity lab, and to be able to offer it to physicians to be used for clinical decision making, we have to validate every sample type. So if you wanted to send me sputum from a patient with bronchitis, I have to have a validated test for that sample type. So right now we're pretty much validated for a broad range of samples, from tissue, blood, urine, semen,
Do you guys do eye
foam,
infections?
eyes. We're swabs
Thank you.
from yeah. So it's pretty hardware. We get
Oh yeah.
metal components, screws from joints that are sent in. So heart valves, we were validated for those, and we just recently validated a test for when one part of the workup is histology. They're going to look under a microscope, and they're going to put the sample into paraffin wax, and so they can look at it under a scope, and now becomes a challenge to they see something, but they don't know what it is. So let's send it to MicroGen and have them sequence it and see what's in that sample. Well, that's paraffin, so we have to validate how to get the DNA out of the wax. That's hard.
It's funny
That's
that
a
the
broad range.
infectious disease doctor you guys had me talk to specialized in joint, like titanium joint infections, stuff like that. He's involved in studies with orthopedists, and so he's a really interesting guy. You and I really hit it off. It's kind of fun because our biotech has done some work in that area. Okay, because I better get to the next question. Do you have any patient testimonials on any of this? I have tons of them. Do you have any? I'm sure you guys have thousands.
Oh yeah, I mean, it's so frustrating bringing this to human medicine with a lot of naysayers and skeptics, and so, what keeps me going is the patient testimonials,
Yeah, they're
the
good.
success stories, and we had a police officer call our customer service center, and he was just calling to say he wanted to thank us and for saving his life, and so how did we save your life? And he said, well, I was diagnosed with chronic bacterial prostatitis. And obviously that's, is inflammation of the prostate. And, he was treated with this typical, we think this is a gram-negative bacteria, so E. coli is causing it. So here's Cipro for 6 weeks. And he had been on one round of antibiotics after another, and it was not helping. And he was pain constantly. And this went over a year like this. And he said he didn't want to live his life anymore in chronic pain. So he was gonna kill himself. And so he went to a urologist that used us and knew about our technology and sent in a semen sample and got him on the right antibiotics. And he told us, he said, I'm symptom free now. I mean, I have my life back. And it's all because of your technology that I was finally diagnosed correctly and put on the right antibiotic. And he was, just grateful. And it's like pretty powerful that people live in misery and pain, and then just say, I don't want to live anymore.
Yeah, me and my sinus infection can attest to that. This just went on, and yeah, I can relate. I get patients like that all the time. I had a patient come up from South America with a testicular infection. No one could figure it out, and we used your technology. We found really weird bacteria. I don't know where they came from. I never asked, but I wanted to. But and we were able to clear it up. And the guy was in agony all the time. I don't know how they got in there. I don't want to know, but we got rid of them. And yeah, a really interesting case. I don't know how you guys had the library for those. Shocking, because I'd never heard of them, either one of them. So
oh yeah, I mean,
saved his life.
if you just look in urine samples alone, we have more than 200,000. And take a guess on how many bacterial species we find in people's urine.
On average?
No, in just in total number of species we've identified in urine.
Oh gosh, probably 100,000.
No, not that many. 6,000 species.
Oh, that's a lot.
Different species of bacteria and over 1,000 fungal species.
Oh wow.
Yeah.
Yeah. No telling how they get in there. We don't want to know. So I don't want to ponder that one. That's too horrible to contemplate.
Well, there's actually an interesting case that made the front page of New York Times in New England Journal of Medicine. It was kind of the landmark case that put next-gen sequencing out there. It was a 14-year-old boy who went to the Caribbean with his family, and he went swimming, and he came back to Wisconsin, and he had headaches, and he had multiple ER visits. Eventually, he ended up in the ICU, and they were drilling holes in his skull to relieve the pressure from the inflammation on his brain. He had massive diagnostic workup. They couldn't figure out. They had him on broad spectrum antibiotics, antifungals. Nothing was working. They took some cerebral spinal fluid. They told the parents, well, there's one last thing we can do. We're going to take some cerebral spinal fluid from your son, and we're going to send it to a lab that can do next-gen sequencing. They got the result back. We weren't the lab. It was the University of California that did the sample. But they found a bacteria called leptospirea. And the antibiotic that only works with leptospirea is high-dose penicillin. So the minute they found the species, they switched off all the big gun antibiotics, put him on high-dose penicillin, and saved his life. And, you think, where do they come from? Well, that comes from animal feces in the water. And he was in the Caribbean and was exposed to it. And that was the start of it.
That's a horrible but great story. Man, yeah, don't swim in the ocean. I won't say that. It's too fun. So, why use MicroGenDx over culturing other molecular tests? I mean, we kind of outlined all this, but maybe you can sum it up for us.
Yeah, I mean, I tell physicians and warn patients that their providers that you grew up with culture, medical school. This is what you learned, and so you're going to go with what you learn and know. And then when it doesn't work, you're going to go to our test. And so when the cultures come back negative or mixed flora and no diagnostic help at all.
Yeah, most of them come back like that. Yeah.
And I tell doctors, you know what? Start that way. Give us the patients that you're scratching your head, you can't figure out what's there, and then send it to us. But eventually you're going to say, you know what? I don't get any useful information from the culture. I'm just going to use the MicroGen test. So it's kind of evolved, process that they have to get comfortable with it. They have to use it in their complex, difficult cases, and then eventually. For my family, I would never want anybody I know to use culture. No, you're going to use our test to get an accurate answer as to what's causing your infection.
Oh, it's light years ahead. It's the Star Trek of testing. It's so cool. So all this technology sounds expensive. I know the cost of it, but what are you allowed to say about the cost? I think it's cheap technology for saving someone's life. I mean, it really is, but I don't even know what it runs across the country. So,
yeah, we're the only lab that actually went to the AMA and CMS and actually applied for our own reimbursement code. Anything in medicine, you want to get it paid for. Wow. And so you have to go through a process. And part of that is having a lot of published evidence. So we have more than 70 studies. We presented that data and they gave us a reimbursement from Medicare for $350. But patients, we basically, if their insurance doesn't cover it, we discount it down to $249. For years, we had it at $199. And frankly, I did that because I got so tired of physicians telling me, well, culture's cheap, Rick. So I'm like, well, how cheap do you want this before you'll start using it? And they said, well, if you could get it around $200, because if you walk into a LabCorp request and say, I just want an aerobic culture, and that's only going to tell you the bacteria that are aerobes, that's $75. If you want anaerobes, the bacteria that don't need oxygen, that's another $160. If you want fungus, that's another $400 to $500. So our test at $249, getting you everything, is cheaper than the traditional old way.
Yeah. Oh man, I totally agree. That's such an easy sell for me. I'm just like, it's $249. It's worth it because most of my patients have been to 6 other physicians for this infection, and we'll talk about one of those here in a minute. But it's a nightmare for them. They're sick of being sick. $249 is nothing. It really is. That's a super cheap price. But again, the immense technology you guys bring to bear. So it's really cool. Thank you for doing that. Now I know why you do that. So it is possibly covered by insurance. I don't deal with insurance. I haven't done it for 20 years. I've gotten away from it so long ago, but if they can find a doctor who does it, they can get it covered by insurance.
Depending on their insurance, like anything, if they have traditional Medicare, yes, Medicare pays us. There's no cost to the patient with traditional Medicare. But if you went and signed up for the Medicare Humana Advantage plan,
forget it.
Thinking you're going to get, protection for eye, vision, and other things, Well, they're not going to cover it. And they're legally supposed to cover it, but they don't. So they'll deny it and they'll make us jump through hoops to try to, send in medical records. They'll just make it really difficult for us to get paid. And so we do that for many of the Medicare Advantage plans, but it's frustrating. The commercial plans, it really varies. I mean, Texas Blue Cross and Blue Shield, they cover it. United often covers it, but again, it depends. If you have a great plan with out-of-network coverage, it'll probably get covered. But if you have the low-tier plans with high deductibles, then they're probably not going to cover it. And so it's a shame. I mean, that's why I like doing these. I think the more patients can be aware of this and can go and fight for this, that's what we need. These insurance companies, it's all about profit and denying, which I understand. Physicians like yourself say, you know what, I'm not going to deal with it. I don't want to
My
deal with
patients
the
know
insurance.
that. Yeah.
Yeah.
Well, I just shoot straight with them, and here's the cost, and it's a good value. And this is a great value for some of the things I found. It's incredible. So even for me, it was a great value doing it 3 times, peeling that onion. And I explained to someone we're gonna have to peel that onion and get ready. So yeah. So why don't Quest or LabCorp? Why don't they offer it? I mean, they don't
Well,
offer a lot of stuff, but yeah. But why don't they offer this? This is crazy.
it's almost like the traditional culture market of growing bacteria, even though we know it's highly ineffective, that most bacteria won't even grow in those petri dishes. And that's from 1870s. I like to jokingly tell doctors, like, what other diagnostic tool are you using from the Civil War era? I mean, or you take a roll of Kodak film and throw it on the desk and say, hey, when's the last time you used Kodak film? It came out about the same time as cultures did. And like, you don't use that anymore. You use your iPhone, right? So the whole concept of buying film. But Quest and LabCorp, that market of traditional cultures is about a $7 billion market. And they make a lot of money doing traditional cultures. So the end result is patients suffer because they don't get their doctor doesn't get a good, accurate diagnostic test to help them make the right decisions on putting you on the right antibiotic to solve your problem quicker. I mean, I was in a conference room with Blue Cross and Blue Shield of California last year, and I said to the medical directors, I said, you know what, let's just talk about women with urinary tract infections. They're coming into your providers and you're paying for the visit, and then you're basically ordering a culture. You're then, probably paying for the antibiotics, and then they come back. And now you're paying it all over again. So you're going to pay for another office visit, another round of cultures, another antibiotic. I mean, it's the definition of insanity. I mean, you keep doing the same thing over and over again, expecting a different outcome. And so, hey, you know what? Just if the first round doesn't work, then don't keep repeating yourself. Send it to us.
That's what I do too. When these patients come in though, and some are just looking for me to be their primary care doc at the moment, even though I deal with other issues usually. But they'll go, the Z-Pak always worked. I'll go, we could try it, but if you're not better in 3 days, you're going to come back and get this, or we'll send it to you. We can mail it to you. So, and so here, take one with you. And if you don't use it, just send it back to us or destroy it. And we'll remove we'll add the charge if you do something. So I try to make it easy for them. And then in 3 days, they're going, no, I'm worse. I'm like, swap away and let's see what we get. So, and then it finds it and we're able to cure them. So, it's just a great option. So, how many doctors are using this technology now with you guys? I know you've grown substantially, you're getting more popular. Obviously, I'm not the only one that's heard about this. Do you have any idea how many doctors use it across the country?
Well, we have I think about 8,000 who've used it. Now, the problem we have is the physicians, they'll use it, but then they get tired of fighting the system. They get tired of fighting the fact that their patient's insurance companies maybe not covering it, or the patient didn't want to pay for it. And so, the physicians will say, you know what, we're not going to use it because there's too many barriers to using it. Or, LabCorp and Quest have a lock on the EMR system. So it's really, you're forced to see a lot of patients and to, pay for your overhead and make a living. And so it's easy to go in your EMR system and punch in, and order a culture from LabCorp. And then the results get back in the EMR. So for us to get in the EMR systems, those companies charge us a lot of money. So those are the things that the doctors say, I really want to do this, but it's just so difficult to get it implemented in my practice. And so we need to overcome those challenges. But the biggest advantage comes when change comes is when the patients say, you know what? I want the MicroGen test. I know it might be easier for you to order a culture because you have it in your EMR system and the box is right outside your front door. But that didn't work last time. I want a MicroGen test.
Interesting. When we're done, I'll give you I'll give you three large groups of physicians you guys need to be marketing to. They may not realize who they are. And we can talk about that later. So I'll send it to your staff to look into those, because these are all big cash practice groups. So, and they're quite large.
To your point about physicians knowing about, I was up at Columbia Presbyterian in New York, and one of the residents there had done some work and used our test and some research, and she presented the results at a grand round presentation. And the chairman of the department said, "Where did you get this?" And she said, "Oh, it's a lab we use out of Lubbock, Texas." And she said, "I want to hear about this." So anyway, she had emailed me, and I went up to speak to her, and she's the division head of plastics at Columbia, and she said to me, "Why don't we know about you? This is amazing." She said, "I want this
to govern."
So I said, "Well, we're a small company. I don't have the dollars of a Pfizer to be able to."
One does.
Yeah. And so we're trying to our best to get the word out, and but she said, "Yeah, you need to attend our meetings." I said, "Well, you charge a lot for your meetings. I mean, I just to set up a booth, I got to pay like $5,000, and and then will your physicians even come by to listen? I mean, so." Those are the things we're trying to overcome to get the word out. But things like this, I think we should do a lot more of to help get the word out.
Oh yeah. I mean, I especially if we and we'll push this if you guys let us, like on TikTok and elsewhere, we'll get a big look at it. Yeah. If you haven't hit that TikTok space, that's kind of where it's at right now, which is really weird. I don't even know what TikTok is, but we have a bunch of college kids and stuff who know exactly what it is here. So
we had a company called Evie come to us. They do women's vaginal microbiome, and they said, they're just a marketing company. They just said, well, we want to use your lab to run the samples because you're a cap and CLIA validated test. And I said, okay. And I said, how many samples do you run a month? They said over 1,000. I was like, really? How do you get the name out there, and how do you promote this? Do you have a sales force? She said, no, we use TikTok. I'm like, you just use TikTok? I'm like, well, I'm 62, so I'm not a big social media guy, but I'm learning that that's the future.
Yeah, me too. Facebook's been where it's at; it still is, but TikTok is massively grown, especially with the younger adults. Yeah. So how can my fans and followers spread the word, tell more people about this? What's a way they can do this and not necessarily involve me? Tell their grandmother or their aunt or their cousin to go get this test. How do they do that?
One, I think you just go to MicroGenDx.com and they'll see the patient testimonials, the people that have had their lives have been changed and impacted by our test. We also have a nice animation video that we did that explains to the patient perspective on what is this, and why doesn't my insurance or LabCorp do this. It kind of covers a lot with what we've been talking about, but it's a little long but I need to make a shorter version. But it's a good patient-oriented animation video that's on our website, and that I just actually had all my employees share it with all of their friends and family. And I said, we have 140 employees. If everybody shares this with people they know, and it actually worked. I mean, we had one of the people called me and said, hey, I share it with all my friends and family, and I already got 3 calls asking for how can their daughter get a test kit.
Yeah. What about putting it on TikTok? Can you do that?
Yeah, I would love to do that.
Yeah, you guys should look into that. You need
Yeah.
like to hire an 18-year-old. It's funny because my daughter's 18 and she works for us up in the biotech, and she's the one who's kind of got us into TikTok. She's 18, what? But they get all that stuff. So yeah, if you have a daughter that or a neighbor or someone who's 18, they might be able to do that kind of work for you.
I'm missing that because I have a 19-year-old daughter, and she said to me, Papa, none of my friends know what MicroGen DX is. So
Yeah,
I
you
said,
can do this virally on TikTok and spread the word. It's a cool test. It's a
yeah.
great test. Okay, good. Let me see. I have this list of questions. Well, if you want to ask me any questions, now's your chance. So
Well, how did you first learn about us? Was it a
I
patient that brought it to you or
I think so. I think
colleague?
it brought me a result. I was like, this is really cool. What is this? And then I got to digging into your company and thought, wow, this is a really cool technology and test. Your library then was about 50,000 strong. So it wasn't that long ago. And now I just did some Facebook Lives about it. Now I get people just order them off our website and use them, and then I have an appointment with them. And we get probably 4 or 5 a month, maybe. So it's not a ton, but because I don't really do much infectious disease work. But
Okay.
but these people have been to my average patient's been to about 15 different doctors before they end up with me. They're
Yeah.
searching for the right answer, and I deal in root causes. So that's my deal. So, and a lot of times they're fatigued because they have a chronic infection somewhere that no one's been able to figure out. They're exhausted from it. And I did a lot in interstitial cystitis cases. We did a study for our company on one of our products for interstitial cystitis, and before we could enroll people in the study, we had to clear them from any infections. We used your technology to do that. So, and guess what? We actually cured several interstitial cystitis cases. Um,
I, I was going to tell you that we actually have a major study going on in IC interstitial cystitis with the top thought leaders at Wake Forest and UCLA and University of California, San Diego, and they were all skeptical of our technology. And
yeah.
if you look at the criteria to be put into this category of IC, these poor women, if they get two negative urine cultures, they're like, oh, it's IC. It's like it's the most ridiculous
Yeah.
diagnostic test to put them into this diagnostic criteria of IC. In the study, the first 40 patients, every one of them had negative cultures. We found known neuropathogens in every single one of them.
Yep. Yep. That's what we probably resolved 80% of the people who applied for to be in our study. We resolved their IC, trying to get into the studies. They like that, by the way. But
Yeah.
yeah, they need to tell their friends about it. Yeah, no, that's kind of how we got into it. And now we really like it. Now the patients just know about it and just order it. So sometimes I don't even know who's ordering it. Then I see him 2 weeks later with the results. So and if
Just
it's
so
go
you
ahead.
know that in the IC patients, because they've often been hit with the wrong antibiotic and they'll have resistance, they're also more likely to have a more of a complex community established in the bladder wall. And
Oh yeah.
now the typical 7 days or 10 days of antibiotics is not going to kill it. I mean, I tell patients all the time, we can help tell you what's there, but you still got to kill them. And so they're much more difficult to kill. And you might have to use multiple rounds of antibiotics. I mean, some physicians are having great success in taking those antibiotics and put them into a insulation or irrigation and putting them into the bladder. That way you're delivering literally 5,000 times the concentration at the site. And so those are the types of things that you have to start looking at with these patients that have had these infections for a long time.
Interesting. Again, it's kind of like when I peeled the onion of my sinus infection. These women with IC, you kind of got to do that with some of them too. You're like, okay, that's round one. We killed 5 different bacteria. Now you're having symptoms again. Let's see. And then you find out they have a fungus in there and 2 other bacteria, and you're just like, God, yeah. So, but you get rid of it eventually, and then they're like, I haven't had pain in 3 months. I'm like, okay, you're there. So
Yeah.
praying that it doesn't come back. You know what to do if it does. I'm here. So,
Yep.
just call us or order one of the MicroGen. So yeah, no, so how many lives has your technology possibly saved? You guys are such a cool company. Do you have any idea? I mean, I can name several dozen on my list who've had infection script that they've had for years, and they was killing them. So
Yeah, it's hard. We don't always get, even in our clinical trials, we don't always get the final outcome. People, you lose them to find out. And a lot of people, obviously, I'm better, so I don't want to go back. And so you don't know. But I know there's been many cases where, I mean, we had a woman that had was a transplant patient for kidney, and she had failed and lost the kidney from an infection, and she had developed a wound abscess on her leg, and they was being treated by a wound care doctor who used our technology, sent a sample in, and then when the doctor got our report back, she said, "Oh my God, it was a rare fungal species or mucor, which will kill you." And so typical transplant patients, they're on immunosuppressive therapy, they're susceptible to fungal infections, and the wound care doctor called the transplant team and said, "I found the reason why she lost the kidney, and she has a rare fungal species." And he sent them the report, and the transplant surgeons were like, "We got to get her in the hospital now and get her on the right antifungal." But they told her, they said, if they hadn't found this, you would have been dead.
yeah. I had a lady come in. She actually was a distance telemed call, and she'd been to see 6 different urologists, and no gastroenterologists. She'd been to see 6 different gastroenterologists over about a 2-year period. She traveled to South America, come back. No one could find out what was going on with her gut. Why she had this horrible diarrhea all the time. She had lost about 40 pounds. And we did the MicroGenDx swab on her. She did it, and then we sent it in, and it grew out a pathogenic E. coli. I was surprised she was alive, and we were able to treat it, clear up. And she, every once in a while, she just calls to say hey and thank you, and thanks me for. She used to call you guys, not me, because
Yeah.
she figured it
Yeah.
out. It was a pathogenic E. coli, and they never found it. I don't. How do you not find that? I don't even. Yeah, I mean that's just a nasty infection. I had another lady, 78. Her daughter called me and said she keeps getting these bladder infections. There's all this pain on her right lower side, and so we did a MicroGenDx. She had nothing in her bladder, no bacteria, no genetics, nothing found. And I said, "Is it so? It's on the right side, low down." And she said, "Yeah." And I said, "Does she have trouble walking?" She said, "Yeah, because it hurts so bad. She has a hard time walking." I'm like, "Has she had a hip replacement?" And they said yes. And I said, "You need to get that scan. She's got an infection in that hip, I bet."
Yeah.
So because she had her white blood cell count was about 8.9, but for a 78-year-old, that's too high. You need,
Yeah.
I was just like, no, she's got an infection somewhere. It's either, in someone's mouth, or it's a wound, or it's, in their bladder. But for her, it was in her hip. And it turned out actually to be a cancer in her hip socket.
Wow. Wow.
Yeah. And it gotten infected. So,
In one of the first fascinating orthopedic cases we had, I met with the top researcher in joint infections in the world at the Thomas Jefferson, the Rothman Institution in Philadelphia,
oh, yeah.
Dr. Parvizi. And he was kind of skeptical, but said, I'm interested. And then 2 weeks later, he blew up my phone, and I called him back, and he said, unbelievable. He said, I had a patient that had shown up in an emergency room on a weekend with an inflamed knee. They put a needle in it, aspirated it, sent it off to microlab. Nothing grew. They sent him home on antibiotics. He got worse. His wife took him to a different hospital. Now he was going septic, and so was headed for the ICU. And the wife called Dr. Parvizi and said, "Hey, you did my husband's knee. You need to help him." And so Dr. Parvizi transferred him to Thomas Jefferson, took him to the OR. When in the OR, took 4 samples, sent it to the microlab at Jefferson. Nothing grew. Sent us 4 samples. Every one of the 4 samples grew. We detected Streptococcus canis, which
Wow. From his
he
dog.
was he was floored, because he's like, "Oh my God, this This guy was culture negative in 3 hospitals, and these guys in Lubbock, Texas, say his knee is full of Streptococcus canis, which is a bacteria from a dog's mouth. So he calls the wife and says to the wife, "Well, do you own a dog?" And she said, "Yeah, we have a dog, and my husband had a cut on his leg, and our dog's been licking his leg." So the dog's bacteria from the dog's mouth transferred to his knee and created a massive infection. And only way they would have found it was through our technology. But Dr. Parvizi said to me, "I think you got something here."
Funny. Well, that reminds me of a it's kind of a cowgirl ranch hand that had this chronic sinus infection for about 4 or 5 years. We did a MicroGenDx swab on her. She had 5 different bacteria. I swear they were all from dirt and pig poop. And it's just like, how does this get in your nose? Well, she's out there raking and shoveling, and she's a farm hand, so ranch hand. So I'm like, we're N95 masks now whenever you do that, once we cleared it up. So yeah, interesting. Yeah, the dog. Yeah, dogs do not have clean mouths.
No.
Oh,
Yeah.
yeah, great story. Wow, oh, this has been super interesting to talk to you. The technology is great. It's a lifesaver, game changer. If you have a chronic, if anyone watches this has a chronic, crazy sinusitis you've had for years that no one can figure out, and they're wanting to do surgery and all this other stuff on you, you may want to get this technology or test first and figure out what's growing in there, because it's probably, like me, 4 or 5 different things. You got to peel that onion, but it'll save your life if you need it. Or if you've got a bladder infection, you got interstitial cystitis, and you know it's an infection, which most of these women know. They're like, I keep telling them it's an infection. They wouldn't believe me, that they need to get this technology. And any kind of weird skin infection or boils or anything like that, get this technology. It just does not miss. It's probably the coolest advent in medicine in I'd say in a decade or two. It's a really neat technology. So and I am on the cutting edge in a lot of stuff we do with our intracellular vitamin testing and technologies and other things we use. This is just a great test, and it'll stay in my armamentarium until I die, probably. So hopefully that'll
That's
be
very
20
nice.
years. So thank you so much, and I love you guys. You saved several of my patients' lives, and we much appreciate it. Thank you, Rick Martin and MicroGenDx. We really love you guys.
Well, thank you. That's kind words I greatly appreciate and keep us motivated to keep on doing what we're doing.
Huh.
Thank you. I appreciate